This section on the chapter from the Integumentary System is going to concentrate on the appendages of the skin, as well as considering the structures that are important for cutaneous sensation, namely the sensory endings that we find in the skin. The appendages of the skin are the glands, hair, and nails, things that are an extension of one of the two major layers of the skin. I also want to consider sensory endings here as we're thinking about things that are found within the dermis for the most part. I want to talk about glands and then sensory endings and then go on to hair and nails. Hair and nails aren't terribly interesting or important structures within the skin other than to just demonstrate variations on the structure of epidermal tissue. So we'll look at those for that reason. The glands in the skin are the sudoriferous or sweat glands, sebaceous or oil glands, mammary or milk glands, and ceruminous or wax glands. Now, the textbook only really introduces the sudoriferous and sebaceous glands, but I want to include mammary and ceruminous here as they are glands found in the skin. They're just specifically found in very limited areas of the skin. This image from the book shows the basic structure of the skin, and it points out specifically the eccrine sweat gland, which is one of the two types of sweat glands, which I'll talk about shortly, as well as you can see the glands that are attached to the hair follicle here, and those are the sebaceous glands that produce oil. This micrograph highlights the sebaceous glands, which are attached to the hair follicle. We see 3 on one side of this follicle and 1 partially on the other side. The micrograph also shows us a little bit of a sweat gland. Off to the right side of the micrograph, there are some small darkly stained epithelial walls, and those are around the duct of a sweat gland. Again, sudoriferous glands are sweat glands. There are 2 types. The most common is the eccrine gland, which makes regular, very watery sweat that helps decrease body temperature by evaporating away when the skin is radiating off heat from the blood. Eccrine glands are found pretty much everywhere. There are quite a bit found in the palms of the hand and the forehead, places where we need to radiate off quite a bit of heat, but they are found most everywhere around our body. Apocrine glands are a different type, which we usually think of as the smelly glands, though they really aren't. The secretions from the eccrine and apocrine glands are not in and of themselves smelly, but apocrine glands are located mostly around skin that has hair in it, like at the armpits and the groin and around the beard. And bacteria that live in those areas will metabolize molecules that are found in the apocrine secretions, and that will produce a smell that we associate with sweat. It can mostly be handled by keeping the areas clean and free of bacterial growth. There are certainly treatments for that, which are commonly called deodorants or antiperspirants. Deodorants will mask the smell. Antiperspirants will cause the opening to sweat glands to close up more so nothing can be released, which is not always a great thing to do. Apocrine glands are often associated with reproductive behavior because partly where they're located is related to reproductive function, and they become active with puberty. Before puberty, they're not an active type of gland. There is some suggestions that the smells that are associated with apocrine gland production can influence reproductive behavior. There is a type of party which I have read about past in a New York Times article, but I couldn't find a reference to it on the internet, called an apocrine party, where people would come to the party with a used t-shirt in a Ziploc bag and deposit that shirt somewhere, and then smell shirts of potential partners. And they would be introduced based on which shirts they smelled most attractive. In the article that I read about this, one of the people being interviewed said, "Oh, they all smell. It's just some smell differently than others," and that would lead to introduction. How long-lasting those introductions are is questionable, but there is some interesting suggestion that there's a relationship between the smells associated with apocrine secretions and reproductive behaviors. Mammary glands and ceruminous glands are not highlighted in the book, but I want to mention them because they are actually a type of gland found in the skin. In fact, they're variations on sweat glands, just mammary glands have additional things included in them, such as lactose sugar and milk proteins, and ceruminous glands secrete a waxy lipid substance. Obviously, mammary glands are located around the areolae of the breast tissue, and ceruminous glands are located in the ear canal. I'd like to take a second to make a public service announcement about wax glands and the tendency to try to clean out the ear canal, because that actually works against what ceruminous glands are actually for. There is a saying that you shouldn't put anything smaller than your elbow in your ear. Now, I have a picture here of George Clooney from his ER days because there's a clip from, I believe it's the 3rd episode of the 1st season, where he gives that specific advice to a patient, to a mother whose son seems to have put something in his ear canal. And the other picture that I have here, which obviously is from Northwestern University, demonstrates the problem of putting something like a cotton-tipped applicator into the ear, which can potentially damage the eardrum. Now, I have a video from YouTube that addresses this issue. There's a second video actually that's not in this presentation, but if after watching the first video you just move the presentation over, you can see the second one. It's a infomercial type of commercial selling a implement to remove earwax from the ear, which is not really in the spirit of what I'm saying here, which is don't work against what wax glands are for. But it is quite amusing in that trope of the poor guy that just can't do things normally and his life will be so much better if he just buys this item that he can see on television. It's an amusing thing to watch, but the first video really kind of gets at the issues of why you should be careful cleaning out your ear canal.
Do you clean inside of your ear with a cotton swab? Some experts say that people shouldn't clean the wax out of their ear. A natural process in human ears cleans out the wax without needing a cotton swab. Using a swab on the inside of your ear can cause wax to get backed up and accumulate inside rather than cleaning it out. Also, if the swab gets pushed in too far, it can damage the eardrum. Simon Bayer, a consultant ear, nose, and throat surgeon, said, "Wax is actually beneficial to the ear. It causes foreign bodies to adhere to it, preventing them from going further into the ear, and it has antibacterial properties. Removing it is like taking the wax off the surface of polished wooden furniture." Thousands of people a year seek medical attention for cotton swab-related problems. Some people's ears do produce excess wax that needs to be removed, but for most people, the ear canal does not need to be cleaned out.
While not actually an appendage of the skin, so to speak, the sensory receptors there are very important. And this figure, which is not in the book, but I found online, and there's a link to its source at the bottom of this picture, shows several of the different types of sensory endings. Now, there are free nerve endings, which are usually found right at the transition between the dermis and the epidermis. They often are associated with pain and temperature sensation. They're thermoreceptors and nociceptors, or pain receptors. And then there are root hair plexus nerves, which can sense the movement of hair in your skin as it stands up or if something brushes against it. And then there are a series of different types of specialized sensory endings. We've already met one called a Merkel cell, which is actually in the epidermis, and it's the only structure here that's actually in the epidermis. There are then a few other things called corpuscles, a tactile corpuscle, a lamellated corpuscle, and then what's named the Ruffini corpuscle. And those are specialized cellular structures around nerve endings that help with specific types of touch stimuli. This image suggests the concept of what are called receptive fields, or the patch of skin that a single nerve ending is responsible for conveying stimuli information about to the central nervous system. Anything that touches within receptive field number 1 is sensed as being from a discrete part of the skin. Anything on receptive field number 2 is from a different discrete place. But anything within one of those receptive fields can't be distinguished from each other, because any of those nerve endings ending right there in that sort of halo that's colored in all go to the same nerve fiber that gets to the brain. So the brain can't tell which part of the receptive field was touched, just that little area. Our more sensitive parts of our skin, like our fingertips and our lips, as opposed to less sensitive areas like the small of our back or the skin of our thigh, have different size receptive fields. The smaller the receptive fields and the denser the nervous fibers in that part of the skin, the more sensitive we are. And the larger receptive fields with fewer fibers going to the skin in general is less sensitive. The hair follicle is truly an appendage of the skin. It's an extension of the epidermis. It has some characteristics similar to the epidermis, but in its own way. Similar to the living layers of the epidermis, the stratum basale, the stratum spinosum, and the stratum granulosum, the hair follicle has layers of living cells that are close enough to a blood supply in the dermis to get nutrients and survive. Those are called the root sheaths. There's an inner root sheath and an outer root sheath highlighted in this picture. Then there's the hair itself, which is actually dead cells, dead keratinocytes. And there's three components to that: the medulla, which is the middle of the hair shaft, the cortex, which is the outer part of the actual hair, and then the cuticle, which is cells that kind of cover up the outside but usually flake off not too far from the part of the skin where it comes out. Now, there are shampoo commercials that will tell you that you have to buy their expensive shampoo to keep the cuticle cells healthy and happy and that sort of thing. But they are dead cells. There's not so much you can do to keep them healthy past the fact that they're dead. Now, hair color is based on pigments that are associated with these cells in the medulla and the cortex. And as hair goes gray, it's because the cells in the medulla stop getting their pigments that give it its various colors. At the base of the hair, we can see a matrix, which is where the cells actually are that give rise to the hairs and the hair cells themselves. That's equivalent to the stratum basale. That's where the mitotic divisions are taking place. There's a dermal papilla, or a little bump of dermal tissue that has blood supply and other components that get into that part of the hair follicle to provide it with nutrients as need be. If you've ever seen a CSI type show where they're looking at DNA evidence from hair that's been recovered from a hairbrush or something like that, what they're actually looking for is hair bulbs, that part that contains the hair matrix, that are still attached to hairs that might be in the hairbrush. And from that they can extract cells that had been dividing and do testing on DNA. Nails are perhaps even more like epidermis in their structure than a hair follicle. Instead of the stratum corneum or stratum lucidum, there's just the actual nail itself, which is again dead keratinocytes. The cells directly underneath it are the living layers of the cell, living layers of the skin, the stratum basale, stratum spinosum, stratum granulosum. The actual place where the nail is growing is at the nail root, and it pushes cells out in the direction. I'm sure everybody's familiar with nails growing. Now, something important about hair and nails together is what makes them different from the skin, besides some structural differences in how they're put together, is also that they contain a different type of keratin molecule. There's still keratinocytes in there. They're just making a stronger type of keratin, which keeps the cells together better, and so the hair or the nail itself doesn't slough off like the outer layers of our epidermis do. We have to physically cut them usually. Now, hair sometimes will fall out, but the entire hair will as the follicle stops growing. The nail will eventually get brittle as it grows out fairly long, but just the same, those keratinocytes hold together much better than the keratinocytes in the stratum corneum of your typical epidermis. Consider this question. When you feel that you've reached an answer, hit the next button to go into the next slide, which will reveal the correct answer. Of the four types of glands, sebaceous glands are the ones that secrete an oily substance. They're also slightly different from the other three. Sudoriferous glands, producing sweat, mammary glands producing milk, and ceruminous glands producing the waxy substance called cerumen, are all generally related types of glands, and sebaceous glands are very different, and they secrete an oily substance from the hair shaft, covering the hair and then getting out onto the surface of the skin, helping to keep water in, as water can't evaporate through an oily covering.